Thank you, Mr. Chairman. Let's see if I can have my presentation shown in the screen. Okay, as you all know, life is a complex phenomenon. First of all, it exhibits structural complexity, which expresses itself at various hierarchical levels: monomers, polymers, supramolecular complexes, organelles, etc. The cellular architecture shows a remarkable stability despite the constant turnover of the molecular constituents. Life also possesses organizational complexity. There are intricate networks of interactions with multiple connections and feedback controls. These include metabolic pathways, networks to regulate gene expression, signal transduction pathways, etc. Organization is also hierarchical. There are networks of networks. And life is also complex in what I call its spreading out or dissemination. In this case, I cite as examples processes such as cell differentiation, morphological development, the ability for niche construction, the dynamism of evolution, etc. Now, going back to structural complexity, what would be the simplest cell we could think of? And to answer this question, I borrow this figure from a paper published by Shostak, Bartel, and Luisi in Nature about 10 years ago. This theoretical cell would consist of a vesicle that contains a ribosome that copies itself and copies also another ribosome which is involved in the synthesis of the membrane, thus closing the autopoietic circuit. This minimal cell possessing only 2 genes or 2 ribosomes would exist, of course, in an RNA world. Do we find anything like this in nature? No, we don't. Life is not as simple as this. An appropriate way to look for the simplest cell is to identify the simplest genome. The minimal genome that has been able to obtain after knocking down genes one by one in mycoplasma, which is the smallest organisms capable of autonomous growth, is composed of 430 genes, 387 genes encoding proteins, and 43 encoding RNAs. Of these, about 100 have an unknown function, although they are essential because they cannot be knocked down. So what you see in this slide is a map of the simplest cell based on the genome, although we still don't know what 100 genes are doing in the cell. Just to give you an idea of how complex a cell can be, it has been calculated that each E. coli cell contains 100 million metabolite molecules and 2.4 million protein molecules. From this, I think you can extrapolate to the complexity of a eukaryotic cell, which is about 2,000 times bigger than a prokaryotic cell. Now, with respect to metabolites, or the molecules of life in general, which is the subject of this topic, they are very characteristic. We know that. Amino acids, sugars, nitrogen bases, lipids, proteins, polysaccharides, polynucleotides, and so on. Although we know they are not exclusive of living beings. Indeed, analysis of meteorites that have fallen into Earth have shown that they carry a wide variety of organic molecules similar to those that are present in living cells. Here I am showing the Murchison meteorite which fell in Australia in 1969. A couple of years ago, a paper was published in PNAS to summarize 40 years of analysis of this meteorite, and the authors concluded that extraterrestrial chemical diversity is high compared to terrestrial biological or biogeochemical spaces. So, presumably, the availability of organic molecules would not be a limiting factor for life to develop elsewhere in the universe. There was a report in the press a couple of years ago about DNA being found in meteorites, and it was not actually DNA, but the nitrogen bases, which is still very impressive. Now, one may ask, would life be possible with a different biochemistry? This question is difficult to answer because we know of one type of life only, but there have been efforts to envisage life with other material components. Perhaps the most consistent work in this area is a study conducted by the National Research Council in the US, which issued this report that you can obtain in the internet, in which they speculate about life with different biomolecules and even with solvents other than water. However, most authors agree that the biomolecules are the most appropriate for life as we know it, which implies that carbon is the best atom for scaffolding supramolecular structures, and water is the best solvent we could think of. A statement made by the prestigious microbiologist Norman Pace in this seminal article entitled The Universal Nature of Biochemistry represents well this viewpoint. I quote, "Indeed, it seems likely that the basic building blocks of life anywhere will be similar to our own in the generality, if not in the detail," end of quote. Similarly, the Australian biochemist Michael Denton, who wrote a book called Nature's Destiny just to argue in favor of this hypothesis, concluded that, and I quote, "I believe the evidence strongly suggests that the cosmos is uniquely fit for only one type of biology, that which exists on Earth, and that the phenomenon of life cannot be instantiated in any other exotic chemistry," end of the quote. This may seem a rather anthropocentric observation, but all remains speculative until we find life in a place different than Earth. In any event, there are some characteristic features of life chemistry that one would expect to find in any living organisms in the universe. They are listed in this slide. First, life is chiral, then the electron transport chain, and the metabolism genetics duality. I will now briefly refer to each of these properties of life. The concept of chirality applies when an object and its mirror image are not superimposable. A textbook example to illustrate this situation is that of hands. In biochemistry, chirality arises when a carbon atom binds to 4 different chemical groups. There can be 2 types of configuration or spatial distribution of these groups, one being the mirror image of the other. These are known as optical isomers. Life on Earth chose only one of the optical isomers for sugars and amino acids. In contrast, analysis of the organic molecules found in meteorites shows the presence of both optical isomers. The reasons why life chose a particular isomer in each case is unclear, and the mechanisms involved in this selection are the subject of intense studies. It could well be that elsewhere in the universe there is life based on the same biomolecules, but of opposite chirality. The second characteristic of life that I recently mentioned, the electron transport chain, relates to bioenergetics. The laws of thermodynamics tell us that the high organization and complexity of life cannot occur spontaneously. The increase of the internal order, which is compensated with an increase in disorder of the environment and dissipation of heat, is made possible thanks to a constant flow of energy. We know that there are 3 energy sources that can sustain life: sunlight, organic molecules, and inorganic redox reactions. Whatever the source of energy, all living beings follow the same strategy to capture and store it for later use. This consists of a flow of electrons from levels of high energy to a lower energy, which produces a proton gradient whose natural tendency to balance culminates in the synthesis of ATP. What you see in this slide in this figure it's a membrane in the cell with the various proteins that transport these electrons until they are received by an electron acceptor. The formation of a proton gradient due to the flow of electrons activated by solar photons or redox chemistry is undoubtedly one of the most unique and ingenious signatures of life on Earth. It is the fundamental support of the distinctive autonomy of living beings, which is permanently sustained in disequilibrium with the environment. The third very distinctive characteristic of life is its dual character. It is metabolism, the cycles of reaction that you see in the left, and at the same time, it is also genetics. It is very difficult to visualize which started first and how they became interdependent. The discovery of the structure of DNA and of the mechanisms by which the information stored in it is expressed brought great hope that life could be explained exclusively from its material components. But molecular genetics, which originally appeared so promising to the reductionist approach, has been the source of findings that instead of facilitating this task are making it more difficult. Some examples of these difficulties are the finding that in eukaryotes, the great majority of the genome is composed by sequences that do not encode proteins. Then we have the G-value paradox, which tells us that there is no correlation between the complexity of organisms and the amount of protein-coding genes. The phenomenon of RNA editing, still not well understood, by which the coding sequence is modified after transcription of DNA into RNA. Epigenetics, which involves the temporal regulation of gene expression by chemical modification of DNA and histones. Pleiotropy, which denotes gene accomplishing multiple roles. Non-coding RNA, which refers to a large variety of non-protein coding RNAs that were previously unknown and seem to play a key role in regulating gene expression. This is perhaps the area of greater development in contemporary genetics, the non-coding RNA. And I close this list with one of the biggest challenges genetics is facing today, to establish a correlation between genotype and phenotype. The latter, as I will illustrate in a minute, is becoming much more difficult than originally thought. I want to say a few more words with respect to the first and last points in this list. First, the fact that sequencing of the entire genome of several eukaryotes revealed that most of the genome does not encode proteins was very puzzling. What is very interesting in this respect is that Professor John Mattick from Australia has found that there is a direct correlation between the amount of this non-coding DNA and complexity. In this case, he measures complexity as the number of different types of cells in the organism. What you see in this diagram is the ratio between non-coding over total DNA in organisms whose genomes have been sequenced. As you can see, in the black color denotes bacteria, and then the gray color are very simple protists. The blue color are simply eukaryotes. In green, you see plants. Then in yellow, you see insects. And then in red, you see vertebrates. Where we know that in vertebrates, about 98% of the DNA is non-protein coding. So there seems to be this correlation between non-coding DNA and complexity. We can discuss later what do we understand by complexity in this case. But I am not sure whether there is a consensus in relation to this finding made by John Mattick, but this correlation probably needs to be confirmed. Now, with respect to the difficulties in establishing a relationship between genotype and phenotype, I will illustrate this point with a recent article by Dowell and collaborators in which they compared two genomes of virtually identical yeast strains. They also say that these two genomes are as similar as two human genomes are among them. Okay? As you may know, the yeast genome contains about 6,000 genes. These authors knocked down 5,100 of these genes one by one, and surprisingly, they found that only 894 of them were essential to both strains. So 1/5 of the genome is essential when they knock down the genes one by one. This is by itself a very unexpected finding. But then there are additional 44 genes that are essential to one of the strains and 13 genes that are essential only to the other strain. As you see, it is not straightforward to deduce the phenotype from the genotype, and this issue has great relevance in biomedicine today because understanding the genetic basis of disease is becoming too difficult. The reductionist approach has undoubtedly been very successful for the understanding of some aspects of life, such as bioenergetics, catalysis, reaction mechanisms, regulation of gene expression, and so on. But it seems to be failing in the purpose of understanding life as a whole, as a phenomenon. I would like to quote two very prestigious scientists that have referred to this situation. First, the Nobel laureate Sidney Brenner, who more than 30 years ago was already aware of the limitations of the reductionist method. He said the following, and I quote, "I think that in the next 25 years, we are going to have to teach biologists another language. I don't know what it's called yet. Nobody knows. It may be wrong to believe that all the logic is at the molecular level. We may need to get beyond the clock mechanism." End of a quote. More recently, the American microbiologist Carl Wesse, to whom we owe the tree of life based on ribosomal RNA, in a very illuminating article entitled "A New Biology for a New Century," said that, I quote, "An e-engineering mechanistic understanding of biology does not work because it is misleading and fails to capture biology's essence." End of the quote. As a result of this situation, what we observe today in experimental biochemistry is an increasing tendency to a systems biology approach, in which, rather than focusing into the properties of isolated biomolecules, the interactions among them are studied. It has become clear that it is through these interactions that the new properties emerge that cannot be predicted from the components. System biology is precisely the subject of the following presentation by Dr. Lilia Albergina. So scientists are now constructing interaction maps. In this case, what I show is the protein-protein interaction map in yeast, which is called the interactome. This map reveals the presence of several protein complexes, which in turn interact among themselves. More than 500 of these complexes in the yeast cell have been identified, with an average of 4 to 5 proteins per complex. In addition, more than 400 interactions between these complexes have also been identified. The same kind of interactome has been determined for various types of cells, including human cells and cancer cells. However, just mapping the interactome is not sufficient, because as Sharon and Robinson say in this article, it's difficult for you to see, but what they say is that complexity comes in layers, as they say. Once a protein complex is identified, one must study its localization, its function, its dynamics, and its structure. And within each of these properties, there are many more that have to be solved. For example, with respect to localization, one has to know the cellular compartment, its abundance, tissue specificity, organ specificity, and so on. The same applies for the other properties. They come in layers. This is not a simple task, as you may imagine. The interaction among gene products on a wide genome level using genetic tools have also been analyzed in yeast. In this work, 5.4 million gene-gene interactions were tested, which led to the construction of this map that you're looking at now, in which interactions are grouped by physiological process, for example, DNA replication, protein synthesis, and so on. In other words, similar interaction patterns were grouped together. This slide shows the cover of a recent review article which deals with the networks that are being studied in C. elegans, Xenoradvitis elegans, a model organism that possesses 1,000 cells. It illustrates pretty well the new holistic approach in biochemistry. As you may see, there is already information about protein-protein interactions in this worm, gene-gene interaction, protein-DNA interactions, and so on. And of course, there are many more types of networks: signal transduction pathways, cell-to-cell communication networks, networks established between organisms, and so on. It is very likely that following this more holistic approach instead of the reductionist one, that we will reach a better understanding of life at the molecular level. And I would like to end my presentation with a warning. Application of pure mathematics shows that the number of possible interactions among multiple components increases super exponentially as the number of components increase. Therefore, unraveling a comprehensive interactome in a cell becomes a virtually impossible task. The key to solve this limitation is to identify modules of interactions. This is what scientist Christoph Koch says in the article I am showing that was published recently in Science. And I quote, the discovery of modular, hierarchical structures that capture the behavior of complex systems in a causal manner is essential to speed up solving these challenging problems. This emphasizes the need to develop heuristic methods to discover modules. For if appropriate modules cannot be found, understanding of life will escape us. Thank you very much for your attention.